Welcome everybody to our autophagy themed talks in neurodegenerative disease today. We're going to hear about Huntington's, Parkinson's, and ALS. Our first speaker tonight is Yamamoto from Columbia University.
Oh, thank you. It's very important. Hi, everybody. Thank you for coming. So today I'd like to talk about our work in autophagy and Huntington's disease. Huntington's disease is kind of the whole motivation as to why we even started working in autophagy. Nowadays, we kind of think about autophagy in the context of the adult brain and trying to understand what it does in the healthy versus the diseased brain. So as many of you know, this is a pathway that drives lysosome-mediated degradation. And the 2 points that I always like to raise about this pathway is that, first of all, it's highly conserved, right? From yeast to man, almost every single cell that's on this earth at one point or another can activate this pathway. And then the other point I like to talk about is its immense versatility. The versatility is driven from 2 points. First is the formation of the autophagosome itself. It's a multilamellar membranous vesicle that can range anywhere from hundreds of nanometers in diameter to tens of microns in diameter. And that kind of reflects the vast variety of cargoes that this pathway can degrade, from individual proteins to intact organelles such as mitochondrion to, say, invading microbes. Then, of course, it then fuses ultimately for degradation by one of the most powerful degradative machineries available to the cell, the lysosome, which can degrade absolutely pretty much everything in the cell. And so, the cell is confronted, this variety perhaps, or this flexibility of this pathway makes it unsurprising that this pathway is absolutely essential for life. But having said that, it of course presents a very complex question to the cell every time. That is to say, when in the world do I unleash this pathway and what should I degrade? And it's that question that my lab really likes to focus on in terms of this larger pathway, everything about cargo capture. How is it that the cell decides what to degrade when? And by understanding what it is degrading, we feel that we can gain immense insights into why this pathway is important, how are cells using this pathway to its end, and why is it so important for the various organs within the body. Now, the other reason why we focus on cargo and cargo capture really has to do with a long-standing fundamental interest I've had in neurodegeneration research for a very long time, which has to do with the abnormal appearance of ubiquitinated structures, a very common neuropathological feature across the vast majority of adult-onset neurodegenerative diseases. And as they've been identified, it inevitably raised the question in every single disease, what is the role of these structures, if any? Do they contribute to pathogenesis, or are they just some harbinger or indicator that the cell is going to die? And so this has been a question that's been posed for decades and has been very, very difficult to answer, for the large part because we don't fully understand how proteins and aggregated proteins in particular are handled by the cells and what the general life cycle of these proteins are. So case in point, when I started my work in neurodegeneration, like a quarter of a century ago, unbelievable, that hurts to say, I can't believe. Anyway, a quarter of a century ago, people were saying, oh, these are permanent structures in the cell. These structures are, what we know biochemically is how insoluble they are. And so likely what happens is that they form at some point and then they just persist until the cell dies, for example, right? But something that I learned as a graduate student over two decades ago now, is that these structures are far more dynamic than one might seem, despite the fact that these structures are detergent insoluble. So, and the way we did this was we created a mouse model in which we expressed an aggregation-prone protein, in this case protein that causes Huntington's disease, where we were able to see the clear indications that this protein was accumulating for forming these detergent insoluble structures throughout. And by using conditional genetics, we were able to eliminate expression of this aggregation-prone protein, and lo and behold, those aggregated structures disappeared from the adult brain. And so this was the first indication to us that, hey, wait a second, these aren't permanent structures, these are dynamic structures, and there are cellular pathways available to cells, even in the adult brain, that allows for these structures to be eliminated. And so, subsequent to that, there was a lot of rapid progress, work by David Rubenstein, Ron Capito, through several papers, as well as our group, found that one of the pathways anyway, or the pathways we felt were the key pathways for the clearance of aggregated proteins, was this pathway macroautophagy. And it's unsurprising that the pH in the lysosome reaching pH 2 can indeed have the capacity to break apart these structures, and we can show this in vitro. Now, the way by which people have identified autophagy as for clearing aggregated proteins was largely kind of RNAi-based screens. That was the rage at the time. We were no exception. But it's, just understanding by affecting genetically the translation of genes that are necessary for the driving of a pathway, right? That doesn't necessarily tell you that that pathway per se is degrading this structure, right? You could just be affecting protein homeostasis overall, thereby affecting the turnover of proteinaceous inclusions. And so we wanted to kind of push the study a little bit more, and we went to the ultrastructure. We wanted to say, well, if aggregated proteins are degraded by macroautophagy, then we should be able to see aggregates inside an autophagosome. It's a very kind of logical set of experiments. But what we saw kind of pushed us into thinking, a thought that was already kind of bubbling in our heads. One of the things that we knew about this aggregate clearance paradigm, first in the adult brain, and then we replicated it in stable cell lines, was that aggregate clearance can occur under full nutrient conditions. At the time, macroautophagy was considered a pathway, an emergency pathway, a pathway that degraded cytosolic constituents at bulk in response to starvation. We didn't have to starve the cells to clear the aggregates. Then we started to do ultrastructure and started to look at these cells under the microscope. This is immuno-EM. You can see these have two different scale bars. The vast majority of aggregated proteins that were positive for our marker were these amorphous, large, multi-micron structures in the cytosol. But we also had a subset of structures that were about 1 micron in diameter or less that seemed to be membrane delimited. So is this an autophagosome, right? And that was really the reaction that we gave our poor EM microscopist, Andreas, who worked so hard to get these images. We're like, what is this? Because typically at the time, this is what an autophagosome looked like, at least in my brain. And this is a beautiful image taken by Eva-Liisa Escalinen in Finland. But this was kind of like the classic idea, a multilamellar structure large enough to take up, say, a mitochondrion. But importantly, this was also taking up cytosol. It was a bulk degradative event, kind of gobbling up nearby cytosol in response to starvation. This was didn't really look much at all like what we saw. Right? But if we looked around the literature, there was actually something that looked a lot like what we saw, and this was done by Dan Kliansky's group. And it sounded like what we were seeing. It was an aggregated structure that was taken up by a multilamellar vesicle in the context of full nutrient conditions, no starvation. And here we found, and under these conditions, we were like, hey, it actually looks similar as well. But there were two things. First of all, this was in yeast, not in mammalian cells. And second of all, it wasn't known as an autophagosome. It was known as a cytoplasm to vacuole targeting vesicle, or a CVT vesicle. And at the time, this was considered a pathway that was very much distinct from macroautophagy. But subsequent to that, we have now very much accept that the CVT pathway is very much a form of what people now term as selective macroautophagy, the means by which macroautophagy take up cargo in an adapter-dependent manner, and thereby allowing for the discrete and very selective turnover of defined cargo. It's achieved by an approach used by many membrane trafficking pathways, adapter proteins. proteins. So in the case of CVT, aminopeptidase 1 forms an oligomer and a complex, larger aggregated structure, which is identified by a series of adapter proteins. And these adapter proteins just act to scaffold the growing autophagosome membrane around the inclusion to allow for the well-defined targeting of the structure into the vacuole. Back at the time when we presented our potential that maybe aggregated proteins are degraded in a CVT-like manner in mammalian cells, we got yelled at. But now it's very much well accepted. In fact, there are a myriad of adapter proteins that have been identified in mammalian cells, many of which you've probably heard of, P62, optineurin, and many others. We have been focusing on one in particular, and this was an adapter protein that showed up way back when I did the RNAi screen, known as Alpha, or the autophagy-linked 5 protein encoded by the gene WDFY3. Now, Alpha is a ginormous protein. It's 380 kilodaltons. It was first characterized by Dr. Anne Simonson when she was with Harold Stenmark. And together in collaboration, she and I have found that alpha is really essential for the turnover of aggregated proteins, at least in dividing cells. And to do so, it has this capacity of directly interacting with the machinery involved in forming the autophagosome. It has a lipid binding motif that can stabilize the autophagosome membrane. And it can also interact with proteins involved, other adapter proteins such as P62, that's also implicated in the formation of the inclusion itself. And we think the way it works is pretty much modeling after CVT, the way Kliansky had originally described, where alpha is the scaffold that allows the growing autophagosome membrane to target the aggregated protein and ultimately allow for the formation of the autophagosome around just the aggregate. So this is what we found in dividing cells. And of course, we want to get, the whole motivation of this work was to answer a question that came up way back when I was a graduate student, when the aggregates just went away in those adult brains. Where did they go? Where did those aggregates go? Did they go into an autophagosome to be degraded in this alpha-dependent manner? Well, one possibility, we were thinking, yeah, maybe. I mean, alpha is most highly expressed, actually, in the cells of the CNS. But we wanted to check this didactically. So the first way you do this is you find a very, very brave graduate student who entrusts their life to you for like 7 years, well, maybe 6. And to do a series of experiments. So to do this experiment, what Leora Fox in the lab did was create a mouse model, kind of recreate the mouse model we had created way back when, that expressed this aggregation-prone mutant protein. Forms inclusions in the adult brain, and we can do this with conditional genetics so that we can shut off gene expression using TET regulation, and then the aggregates will go away. So what she then did was create another mouse model and bred it onto this mouse. So this was a conditional mouse model that could eliminate alpha. So alpha is normally expressed, and we can use a tamoxifen-inducible Cre to drive gene excision under temporal regulation. And so 3 years later, after she finished breeding these two mice together, she was finally able to do this experiment. So what she did was, in those mice that were expressing aggregated proteins in the brain, right prior to administering dox to shut off or abolish expression of the aggregation-prone protein, eliminated the expression of alpha and asked what would happen. And lo and behold, aggregate clearance did not occur as efficiently in the absence of alpha, suggesting strongly that aggregate turnover relied on alpha in the adult brain for degradation. Now, one of the things about this observation is that alpha is very specific to the aggregated form of the protein. So if we look for detergent-soluble protein, those levels are unaffected. And so by identifying alpha as a key player in the turnover of aggregates, and specifically the aggregates, we were now poised to be able to ask a very fundamental question that I posed earlier. If we affect the aggregation status within a diseased brain, what happens to pathogenesis? Is there any impact on pathology at all? So to do this, we turned back to basically my favorite disease. Are you allowed to have a favorite disease? Huntington's disease. This is a devastating autosomal dominant, fully penetrant disorder that was characterized for many centuries, but even before, but was ultimately in 1872 with George Huntington's publication on the description of this multi-generational disease affecting families in Long Island came to bear his name. It is a disease that is marked by a triad of symptoms from abnormal movements, which it's most famous for, the choreic movements, to cognitive and psychiatric alterations, and neuropathologically is best known for the loss of the striatum itself. Now, it's caused, all forms of Huntington's disease is caused by the same mutation, a trinucleotide repeat expansion in the very first coding exon of the gene HD. And this trinucleotide repeat expansion causes an expansion of a polyglutamine at the very end terminus of the protein. This is a polymorphic region, so also in unaffected patients, you have anywhere from 2 to 35 repeats, but greater than 40 repeats, you are pretty much guaranteed to have this disease. Now, the trinucleotide repeat expansion has a very interesting relationship with age of onset of the disease. And this is one of my favorite examples of Huntington's disease, as well as other trinucleotide repeat disorders, where you can see the number of codons has an inverse correlation with the age of onset. So the longer the repeat numbers, the earlier you get the disease. And if you look at Huntington's disease, the correlation is really quite tight, an R-squared of 0.97, near perfect. And so this is really something that I held on to for a very long time as a way by which to think about, how black and white Huntington's disease can be. Now, one of the very interesting aspects of this interrelationship can also be reduced down to the biochemistry of the protein itself. So as the codon length of the Huntington gene increases, so does the polyglutamine expansion. Well, the relationship between the polyglutamine expansion and the rate of aggregation is very similar to age of onset. First, aggregation in vitro of this trinucleotide of mutant Huntington starts to occur at about 37 to 38 repeats, very close to the to the repeat length for onset. And the rate of aggregation is exponential, just like this age of onset curve. And I thought that the work by Vonker and ultimately Max Perutz was really very telling and giving some indication of how aggregation might be playing a critical role in this disease. Having said that, if we look at the neuropathology, the relationship is less clear. One of the things that's often misunderstood about Huntington's disease is the aggregation pattern itself. In HD, it's not like every single neuron has aggregates. It's actually quite a rare occurrence. Maybe upward, the maximum 20% of the neurons at any given time have aggregates. So it's not like a massive aggregation disease. Also, one of the things that we find is the aggregation pattern is quite complex. It is not only in the nucleus, it's also in the cytosol throughout the neuropil. It is also not necessarily found in those cells that are preferentially vulnerable. There is no relationship, it seems, from the neuropathological perspective of where the aggregates are and which neurons die. So here again, this complexity with aggregation and pathology. Well, now that we have this alpha protein, however, maybe we can get some insight. So in order to do this, we wanted to kind of ask a nuanced question of asking whether or not you can affect the rate of aggregation, and would that at all, or affect the rate of accumulation rather, and if that at all would have any impact on the disease. And so to do this, while she was breeding those other mice, Leora, together with Chris Johnson and Joan Bosco in the lab, took on another project, which was to deplete the levels of alpha in a mouse model of Huntington's disease. Now, if you're 50% alpha, an alpha heterozygous animal, you live just fine. You're completely normal 2.5 years. Your brain looks great. I mean, you are just fine. You breathe. Everything is good. And so we wanted to know whether or not a mouse such as that, slightly reduced in the ability to potentially clear aggregates in the background of a disorder in which a protein is driving aggregation, would that have any impact? So she took these mice and kind of took a look. Now, the BAC-HD mouse model created by William Yang is a very subtle model. It has a very mild aggregation phenotype. And at 9 months of age, this is what you see, hardly nothing. You see maybe an occasional puncta, maybe some hints of some accumulation. But if you deplete alpha by 50%, so under wild-type conditions, these mice are perfectly normal. But in the background of Huntington's disease, you can say that you can rapidly accelerate the aggregation pattern. So depleting alpha, depleting the ability of turning over aggregated proteins, making alpha rate limiting in this process allows for accumulation to occur more rapidly. What happens with behavior? Well, functionality. Here we look at two different assays, the accelerated rotarod and the open field. So in black are the control mice. You have alpha heterozygote mice. As I mentioned earlier, they're quite normal. This can be seen both in the rotarod as well as in the open field. They look quite indistinguishable. If you look at the BAC-HD mouse model, this is just the Huntington mouse model, we see clear age of onset on the time of the rotarod at about 5 months of age with a steady decline over time. And for locomotion, they become about hypolocomotive at around 12 months of age. Well, what happens in those mice where we've increased the probability of accumulation, we've also, surprisingly actually to me, enhanced the age of onset of this phenotype, now with clear age of onset on rotarod at 3 months of age and hypolocomotion manifesting at 9 months of age. And so here again, what we find is that alpha, by being rate limiting in the ability to turn over inclusions, now we can see a functional outcome suggesting that perhaps the ability of your innate ability to turn over, handle protein homeostasis in your cells can actually perhaps drive when you might manifest disease. This is just to say, again, despite the fact that we have increased aggregated proteins, soluble proteins remain unclear. And this is just from detergent solubility fractionation, where you can see more aggregated proteins and diminished solubility and no change in solubility in the presence when alpha is depleted. So this is in the mouse model, and we have like this complex, all the mysteries of understanding the intact brain and how these genetic interactions might impact the intact brain. We see that alpha, the loss of alpha can accelerate the accumulation of protein, impact disease onset. Now, one of the things, though, about these mouse models is that they're extreme. The number of repeats in the BAC-HD is 97 glutamines, which is actually quite a rare mutation. And if you look in vitro, 97 glutamines has a rapid aggregation kinetics. And so one of the concerns we had was, well, maybe what we're seeing is something that's very much about the aggregate. Maybe this mouse model doesn't really have that much active soluble protein or something like that. Can we find a way by which we can look at this in a more biologically human relevant model? And so that allowed us to collaborate with Zhenyu Yue, who developed this approach that really allowed us to finally answer this question in patient-derived materials. So what Andrew has developed is a direct conversion protocol using a microRNA-directed method. And he can take patient-derived fibroblasts, say from a healthy patient or symptomatic HD patients, and convert them to medium spiny neurons. And we adapted this approach because one of the things that he was able to identify by using this approach was that he was able to capture some key features of Huntington's disease neuropathology that had never been able to be captured by using iPSCs. Now, one of the things that's fascinating about this work was that he found that if you directly convert fibroblasts, say, from a 60-year-old patient, and then turn them into neurons, if you looked at the methylation pattern and you follow, say, Horowitz's work of how, Steve Horvath's work about the methylation clock, that if you do direct conversion, you actually maintain the age. So 60-year-old fibroblasts become 60-year-old neurons. If you take those same 60-year-old fibroblasts and make them iPSCs and then make neurons, they become baby neurons. And guess what? You don't capture protein aggregation, and you don't capture some of the other cytotoxic effects that are associated with this model. So anyway, what's really exciting is here's a very patient-relevant repeat length where we can now create neurons and get aggregation. What does Alfie do under these conditions? And lo and behold, we can enhance aggregation if we knock down Alfie in these animals. This is just some quantification here. So you can see there's an increase in aggregation upon the depletion of Alfie. This is with a patient with only 46 glutamines, but you can see even a patient with only 40 glutamines, we can see aggregation, and that aggregation is enhanced. So what this suggests to us is that if Alfie is variable, you happen to have a mutation, you're heterozygous for Alfie, perhaps what this can do is modify the age of onset of this disease. Now you might be like, what are you talking about, You just told me that the trinucleotide repeat expansion drives age of onset. And yes, this is the case if you take this full range of the repeat codon. But one of the things that I had learned coming up in the Huntington's disease field is that even though that this correlation is very widely accepted, if you talk to the vast majority of clinicians, they'll go, sort of. This is sort of true. And in fact, if you look at the vast majority of cases, so here this is, was redone by Nancy Wexler's group. This is looking at the Venezuelan cohort, where if you look at repeat lengths that are more common for disease onset, so about anywhere from here 37 to 50 repeats, you can see that the age of onset can be quite variable. A patient could be, say, 20 years of age or, say, 60 years of age with the same trinucleotide repeat expansion. A 40-year difference. This is huge, right? And in fact, they expanded upon this work together with the New York Genome Center, as well as MIT, David Hausman's group, to try to identify what are the potential modifiers of age of onset. And they have found that there's significant room for genetic variability. So one of the things that we noticed in this paper back in 2004 was that one of the peaks that they identified was at chromosome 4q21. And so jokingly one day we said to Nancy Wexler, ha ha ha, that's where Alfie is. Wouldn't it be funny if Alfie was a genetic modifier of Huntington's disease of age of onset? And Nancy is the type of person who was like, let's check. I mean, that's like saying like, guess what? What Alfie is a big gene, but it's not like a chromosome. Anyway, so she's like, we're gonna check. And so she mobilizes the forces, and lo and behold, Alfie is indeed a modifier. And the potential variant that they identified encodes for a very modest variation of isoleucine to valine at position 3032. Now, before we knew the identification, we were thinking, oh, well, okay, it's probably going to accelerate age of onset because you have a loss of function of Alfie. But no, what they actually found was that in this cohort, carriers of the variant had a significant delay of age of onset, on an average of about 9 years of age. And we're like, what? Well, first of all, we're recovering from the fact that Alfie et al. Was a variant, but then we wanted to understand what this could be. It's a little bit telling in the actual variation itself, and while we can't, I don't have the full mechanism to be able to lay out for you now, suffice it to say that the isoleucine to valine change is not a gross loss of function, in fact, what it did was change the half-life of Alfie, and we're not fully understanding how this might be. So thanks to the New York Brain Bank and Jean-Paul von Sattel, together with MIT and the New York Genome Center, we were able to have sequenced 100 brains from the bank looking for the people that carried the I3032 variant. This was across different disorders. And what Catherine Croce and the lab found was that patients that carried I3032V had higher levels of Alfie. Okay, so what could that be all about? Well, we got very excited because we already knew that in HeLa cells, if you increase the levels of Alfie expression, you can actually enhance the clearance of Alfie steady state. We had posed the hypothesis that Alfie is rate limiting for aggregate clearance, and so we increased Alfie levels, and just so after 3 days of Alfie overexpression, we can see about 30 to 30% fewer inclusions at any given time. And this study, I don't have time to really talk about, breaks down what kind of structures are turned over, and it seems to be an active process. So, to this end, Catherine decided to pursue this further. Let's actually increase the levels of alpha in the mouse model of Huntington's disease, the genetic complement of the loss of function experiments, to see what would happen. And so she's doing two different studies. One is to allow for expression of alpha from the beginning of time. So you always have higher levels of alpha, mimicking what we think is happening with the carriers of the variants, or you use kind of an inducible approach. We call it the therapeutic aim, where you take symptomatic patients with a full load of aggregation in their brain and increase alpha levels to see if we can derive therapeutic benefit. Suffice it to say, for now, I can only talk about this first study where she's really made some significant progress. We can see a good amount of alpha overexpression in the brain in these mice. And so she started to do some behavior. So we're revisiting the accelerated rotarod. This is in a very aggressive mouse model of Huntington's disease known as the N171 Borchardt model. And here you can see how aggressive it is. Unlike the back HD, you have these control animals already at 2 months of age. The mice have a lot of difficulty staying on the rod. And you can see this slight decline. This is across sexes. So what happens if we augment alpha levels? Well, when we augment alpha levels, the mice still have yet to get sick. We're out at 11 months already, and the mice are still not manifesting phenotype. So this is very exciting to us. We're also looking at what kind of transcriptional changes might be associated with this overall, whether or not we are turning back the clock on changes that the HD mutation is known to have on certain mouse models. So in essence, decreasing alpha levels, I show you can increase aggregation in mouse models of Huntington's disease, accelerating disease onset. And now we have, at least for the behavior, the genetic complement. Augmenting alpha levels can now slow the age of onset of this disease. Of course, what is going on with aggregation in this model? Catherine is doing it now. She didn't do it in time for the talk, but she's kind of very nervous to find the results. She's very excited, and so am I, and we're both scared of the results. Okay, so and so this is really it. Now, before I end, I just wanted to kind of make one last point, and that is simply this: that here we are, we're messing around with autophagy, but I think what was really critical about these experiments is that we're messing around with an adapter protein, right? We're being very, very specific. And I think this is extremely important because, there are a lot of questions that we still have no answers to in regards to the fundamental role of, say, macroautophagy even in the CNS, right? And one of the things that is very, very clear is that while we barely know what macroautophagy does in neurons, and we're figuring out what's going on with that every day, we have very little to no insight on what's happening in other cell types of the CNS, which I would argue are going to be the ones that are more robustly responding with macroautophagy, especially in response to peripheral stress. And so this is just something to really keep in mind when trying to manipulate macroautophagy to try to combat or to answer your questions in the context of neurodegeneration. To this end, we've been sort of peeking at this a little bit, and we have work kind of in revision, so we're battling with reviewers as always. But one of the things we're finding is that macroautophagy in oligodendrocytes is absolutely essential for maintaining the myelin sheath. And if you diminish the capacity for this cell autonomous turnover of the myelin sheath by macroautophagy, you can really disrupt axon function, and you get widespread neurodegeneration. And so you could see how just not even touching the neurons and now affecting oligodendrocytes, by macroautophagy in oligodendrocytes, you can have a widespread effect. Another set of studies that we've been doing that really causes us to be very mindful about how we think about macroautophagy in the CNS is a huge proteomic study that we started undertaking thanks to Project ALS, where we were doing tissue-specific, but now cell-type-specific understanding of the autophagosome proteome. What are different cell types degrading by macroautophagy? And looking at this proteome, this is done by Zhenyu Yue in the lab, it has been really revealing. One of the same physiologic stress felt by an animal leads to a different autophagic response in different tissue types and in different cell types. So you imagine that kind of complexity if you just wholesale affect this pathway across cell types, what kind of messed up impact you might have, and really not necessarily answer the question that you think you are answering. So that's all in my pontificating. Anyway, so I would like to thank the people who did the work. I hope I mentioned everyone. Oh, Icahn was doing the oligodendroglial autophagy. Aveline, the cell-based overexpression of Alpha. Kiki was absolutely invaluable. She did the direct conversion as well as the stereologic analysis. We have strong collaborators, Ann Simonson, Andrew Yu, the Keck, what I call the Keck Foundation project, and their immense support in terms of our work with the variant. The work was supported by NINDS, but as well as the Hereditary Disease Foundation and Huntington's Disease Society of America. Thank you very much. I'll take any questions. Or not. Okay. Shy bunch. Oh, thank you.
When the Wechsler group showed that it was a modifier, how did they do that? Thank you.
Oh, God. I wish Mike Zodi was here. They would know how to do it. No, so basically first it was a GWAS. Where they identified the various peaks, and then they went down and started doing sequencing across. I guess they looked for particular SNPs to see how it would move with age of onset, and they did that iteratively, and then used various statistical approaches, and the region that was most probably associated with a change in age of onset fell in Alpha. Does that make sense? That's my understanding of it, but I might have completely said it incorrectly. Yeah, not my area.
So the Alpha mutations in the patients, do they have any other phenotypes in the mutations that you had, the Alpha, higher stabilization of the Alpha itself? Do they influence any other macroautophagies? Because I'm guessing that Alpha is also involved in the autophagy of other proteins or other processes.
Right. So, I forgot to mention that. Yeah. So the variant itself, it's hard to know. We just started the studies on that. In terms of if we just knock out Alpha in the adult brain, for example, we actually isolated autophagosomes from those brains, and from what we can tell anyway, cargoes are still being properly trafficked by macroautophagy into the autophagosome. So P62 is going in there. Mitochondrion are still going in there. Synaptic vesicle proteins are still going in there. All the different cargoes that we anticipate would be trafficked were largely being trafficked as far as we can tell. So there was no gross disruption of macroautophagy. Could there be some cargoes that were missing? Absolutely. And yeah, we're open to that. And so we'll see. Yeah. However, having said that, mutations in Alpha has been implicated in neurodevelopmental disorders. These tend to be a loss of function mutation, so implicated in schizophrenia, autism spectrum disorder, and other disorders.
Thanks.
Is your lab working at all in terms of autism and I'm sorry, in terms of Alpha and autism, are you at all focused on autism?
So I have a postdoc in my lab, Joanna Dragic, and she's been really working on the role of Alpha in neurodevelopmental disorders with a particular emphasis on ASD. The information I know of ASD is from her. And so this is something that she is hoping to take on further as she expands her own lab, yeah.
Oh, thank you. That's very fascinating.
Have you ever tested direct interaction with Alpha and then Huntington and any EM evidence that alpha is really recruit to that aggregate. And if it's very straightforward, I think it will be interesting to test the mutant alpha, whether their interaction is
Sure.
changed.
Yeah, one possibility with the I3032 mutation, the mutation is in the place of important protein-protein interaction. Having said that, so no, we have not, because we hope now we have finally developed an antibody against alpha. The existing alpha antibodies were no good. They were not specific for alpha. So this is something that we're working on now. We do think there is a direct interaction, but that's only through overexpression of alpha. But no EM studies or anything like this. Yeah.
What's the level of the alpha in your animal model?
We say that again? The level? So on the Huntington mouse model shows no, under baseline conditions, you mean like in wild type mice, do they have like less or more? In normal Huntington mouse model, are you asking if the alpha levels change?
Yes, at the bed level, right?
Yes, no change.
No change, okay. And generally, the Huntington models, in Huntington models, autophagy level is compromised. So does the alpha affect the autophagy, bad autophagy, or when you overspread the alpha in your mouse in your Huntington model, what about the autophagy level?
So this is a complicated question, actually. So, all right. So yes, there have been some reports suggesting that there are changes in autophagy in some of the mouse models of Huntington's. We have not seen this in these mouse models. So that's first. Second, if there were to be any changes, one would imagine it's through or I would think it's through the aggregation of alpha. And the reason why I think it might be complicated is because if we increase alpha levels and we drive aggregate clearance, don't we drive the very few measurements of autophagy with which we have, like autophagosome formation, like LC32, very limited assays that we can do. But having said, given my excuse, I don't know yet.
Do you know the cell type specific expression of alpha? Is it more expressed in neurons or microglia or oligodendrocytes, for example?
Right, yeah. So what we do know is that alpha is highly expressed in oligodendrocytes. It does depend upon the stage. I think it's higher in early stage and then comes down. But in the adult brain, it seems to be relatively even from what I understand at the protein level. I think at the transcript level, there are some differences, but we are running some transcriptomics to kind of get a better sense of that.
Thank you.
Jamie.
Do you have any idea how the specificity of the aggregates would work? And have you looked at specificity for other aggregates and like?
Yeah. So this is, so I guess it's always a point I kind of never really make clear. Alpha doesn't care if it's a Huntington aggregate or a synuclein aggregate or a random aggregate. And so how that specificity is achieved, we don't fully understand yet. We think that P62 plays a critical role in the sequestration and the formation of the inclusion. But beyond that, we don't know what are the specific determinants. The model is very loose for a reason. I think there's some missing pieces. We don't fully understand how it works. So there was an early study suggesting that P62 was required for alpha to evoke its function, but we do not see that. And in vivo, we do not see this. We tried to do it genetically, but it's a little bit difficult because when you lose P62, you lose inclusion formation. And so this has been made it a little bit more difficult to be definitive about that. Great. Well, thank you, everybody.
Should I just go?
presentation?
Yep. All right. Good. All right. Thanks, first of all, for the opportunity to showing our work. So we will continue to talk about autophagy. My lab study autophagy as well as neurodegenerative diseases with a primary focus on Parkinson's disease. Oh, that's this one?
It looks like
Uh,
Does it say who was plugging or something?
It doesn't move the slides. Oh, good.
Okay.
All right, good. Okay, so since I has already talked about autophagy, the processes, I just like to share with you a movie that I made it for autophagy. Really want you to appreciate how dynamic autophagy is. So autophagy, we know, mostly started from the ER and likely the mitochondrial interface, and formed this early structure, the phagophore, and start recruiting many proteins, including autophagy receptors and the cargos. And eventually they also have the snare protein, the syntaxin 17, and a pair with the SNAP29 on the lysosome, and then cause the fusion of the autophagosome with the lysosome. And eventually they fuse, and the lysosome will digest the protein or content inside. So this whole process, as you can see, is really a dynamic, the membrane rearrangement process. It takes about 10 to 15 minutes, so it's really a rapid process. And then this is constitutively active inside most of the cells. So I would like to also talk about very briefly the autophagy signaling, what regulates, especially the early stage, the initiation of autophagy process. And one of very important kinase complex, the mTOR, drives the initiation or the early stage of autophagy activation. So as you know, mTOR is a master of a nutrient sensor, and then when mTOR is inhibited, and autophagy can be activated. And we also know, based on the work of many groups, mTOR kinase can phosphorylate many of the protein, especially in the URK1 kinase, the protein kinase. And this is an essential protein kinase complex for synthesis of autophagomes. And another lipid kinase, the VPS34 kinase in my lab, and then the others has identified multiple components in this complex, namely IDG14, and RBF2 Beclin 1, VPS15. And this is the VPS34 kinase that drives the synthesis of the PI3 lipids, and that's very important for the autophagosome formation. So as you can see, many of these proteins in these two complexes, as well as MBRA1, UVRAG complex, and a TFAB, as well known as the transcription factors for driving the lysosome degradation and autophagy also. And many of the phosphorylation site shown here can be carried out by the mTOR kinase, as well as the URK1. Showing here in the green are those the site that facilitated autophagy process, and those in the red cause the inhibition of the autophagy. So I can see there was an extensive phosphorylation signaling carried out by mTOR kinase, as well as the URK1 and PK and others. And this sort of shows the balance of the signaling and at a basal level. When there is a stress, many variety of stress or nutrient starvation can change this balance of the dynamics of the phosphorylation, and it drives the autophagy either activation as well as the inhibition. So as you can see, this is over the past 10 years or so, there was a really increasing evidence showing there's a complexity of the signaling of autophagy regulation at the very beginning of the autophagy process. I would like to also to summarize what we know so far based on our own lab study and others, the regulation of autophagy in the neurons. So here shows the couple of main point that shows the cell autonomous function of autophagy in neurons. Majority of the work is based on the genetic animal models, particularly the knockout of the essential gene based on the autophagy function and also of overexpression. So the work from my lab and others, including I and then also David Sulzer's group, and we showed that autophagy is constitutively active. Not only can be induced in a high level or suppressed, but just at a base of physiological level, autophagy is constitutively active, turning over many protein cargoes. So it's an important process, maintenance of protein lipid organelle homeostasis, and also regulate many aggregates, proteins, and then many of those proteins are involved in the disease process. Well, autophagy is also compartmentalized, and we have evidence demonstrating that autophagy is differentially regulated in the axon as opposed to the dendrite and the cell body, and particularly in the axons. And we have evidence showing that when you knock out autophagy in the neuron, in the cell autonomous level, the manner, and then the most vulnerable compartment is the axon. The axon sense the loss of autophagy at a very early stage and then become really swollen and then degenerate much earlier than the dendrites and also the loss of the somas. And it also recent evidence showing that autophagy is involved in synaptic activity. There's a couple of really nice publications, including David Sulzer's group, showing regulation of autophagy involved in synaptic vesicle homeostasis, and also autophagy is involved in the learning and the memory formation. But the really interesting part for autophagy is the selectivity of autophagy. As I already mentioned, that alpha is the receptor, and then the autophagy, now there's overwhelming evidence that autophagy has strong selectivity, turn over many protein cargoes, and they are mediated by autophagy receptors such as alpha and then P62 and others. The basal level autophagy is mostly neuroprotective, and as we know that knocking out genes of essential for autophagy will drive the neuron degeneration. So, so this slides, I just want to give you a flavor that is selective autophagy, and in many cargoes, including organelles, protein aggregates, lipids, some structure inside the cells. Also, the pathogens that invade the cells can be degraded by autophagy, and this is really all you can eat. Many of the things, including the protein aggregates, as I already alluded to, and also mitochondria, as you all know, can be also degraded. And the degradation is mediated by a whole bunch of autophagy receptors, including P62, optineurin, and many groups are very active working on this mechanism, and it's a very important the disease, a process that also associated with the disease process. And also the ribosome and the ER and the so-called ER-phage, and then this is also very active area. And there was a very strong evidence that the ER can be constitutively turned over by autophagy. And in our lab also demonstrated in a neuron that without autophagy, many of the ER-like structure accumulated in the axon terminal as well as the cell body. That really shows important role of autophagy for the ER turnover. Well, there are many acceptors yet to be identified. And we know that these receptors are so important for really selective degradation of the cargoes. And then characterizing those autophagy receptors or adapters provided much insight into how autophagy, the basal level, as well as the pathological condition involving the diseases. So what I want to talk today is the two stories. The first one is I will talk about the autophagy receptor, the p62, and some of the rare mutation involving the ALS and or FTD, and how the mutation impair the selective autophagy, the NRF2 anti-oxidative stress response. And I will spend most of the time to talk about a new story and that is still not unpublished. The microglia autophagy cleared the neuron release, the synuclein, through selective autophagy and prevented neurodegeneration. So the selective autophagy, it's a very important concept. I think especially over the past decades also. One of the most important receptor is the p62. This is the prototype receptor as being shown in many physiological conditions as well as the pathological condition. Well, basically, the idea of the receptors is on the one side, they can interact with autophagy machinery through the interacting with the LC3, through the Lear domain, and then on the other side, the p62 or other receptors can interact with the cargos. And then, for example, the p62 can interact with the ubiquitin, the labeling ubiquitin, and then take the shuttle the cargo to olfactome and then degrade it inside the lysosome after fusion with olfactomes. So why we're so interested in p62, one of the very interesting evidence based on the neuropathology that p62 is co-localized with many protein aggregates well associated with the disease. For example, tau, alpha-synuclein Lewy body, TDP43, and also Huntington. And then this is at the beginning, people thought this is just a protein that aggregates, and then what's the big deal of that? But interestingly, there are animal model that either overexpress or reduce p62 in many of these protein aggregates related model shows that p62 indeed can modify, because of this overexpression or reduction, can modify the disease progression caused by those disease-related protein aggregates. This really suggests that p62 is a very important protein for the protein quality control, for perhaps for the protein aggregates degradation, just like Alpha, as I already talked about. So we are interested in the role of a p62 in the neurons, and especially how is that associated with the disease progression. So we are interested in one of the mutation identified previously, showing that through the GWAS study, showing this G425R mutation that in the ubiquitin binding domain of the p62. The p62 has a very interesting domain motif. For example, on the N-terminal, there's a PP1 domain is important for the protein polymerization. And there was a Lear domain that, as I mentioned, interact with the LC3 that associate with the autophagy machinery. And there's a key motif that's important for the KEEP and also NF2 binding. And it's regulating the anti-oxidative stress. So the mutation occurs in the UBA domain, and then through the computational modeling, we can immediately tell this mutation caused the steric clash between the UBA domain and ubiquitin binding. And also our experimental evidence shows that really reduced the interaction based on the ITC assays, disrupt the monomer ubiquitin binding in the UBA domains. And I also want to point out that the UBA domain of the p62 has a couple of very important protein modification site, phosphorylation site, namely the serine 403, and that is phosphorylated by the TPK1, one of the very important kinase that associated with the immune response. And also the mutation TPK1 is associated with the ALS. And there's another phosphorylation URK1 site, the serine 407. And then this is, as you know, this is an autophagy kinase, and it's essential for autophagy biosynthesis. And these two kinases can phosphorylate p62 at both sites, and they drive and increase the affinity of the PCT2 to ubiquitin cargos, and that drives the sequestration of the ubiquitin cargo for autophagy degradation. So what we found that is this disease-associated mutation can really abolish or diminish of the 2-phosphorylation site, and that also reduce the ubiquitin binding of the cargo to PCT2. So just to summarize quickly for you what we found, this mutation of the disease, associated mutation, it disrupts the sequestration and clearance of the ubiquitin protein cargos, and it reduced the phosphorylation, another one at a serum 349, and it is the mTOR phosphorylation that actually changes the anti-oxidative stress. And we also demonstrate in the lab that impairs the interaction of the PCT2 with the KEAP1, and it reduces the NRF2-mediated anti-oxidative response. And this mutation also enhanced the TDP43-associated stress granule formation upon oxidative stress. So in a neuron, when we put back the PCT2 disease mutation in a cultural neuron in the absence of PCT2, I just want to make sure that to say that PCT2 knockout causes no effect for the neuron at all. But when we put back the wild type PCT2, there's nothing going on. But when we put the mutant PCT2, and we found there was a gain of function of the mutation, it caused a much reduced the complexity of the dendrites, and eventually the neuron degenerate. So this is a really interesting phenotype that would demonstrate PCT2 through probably the change of the function, especially the KEAP1 anti-oxidative stress, and it causes the neuron degeneration. So not only in PCT2, a bunch of ALS-related genes that are associated with also autophagy function. One of the most well-known genes, Sinai alpha 72, and there's data from probably 4 or 5 groups, all demonstrate that this protein forms a complex and is tightly associated with URK1, the autophagy kinase, and it changed the URK1 function. And this is really an emerging picture for the Sinai alpha 72 that could be associated with the URK1 associated protein trafficking, including autophagy. And then other genes, including TPK1, alpha-synuclein, and there's another autophagy receptor, the mutation also caused the ALS. So just really, there's an emerging picture. I want to emphasize that. We also propose maybe autophagy, the ALS could be just autophagy disease. And then we propose autophagy is an important drug target for ALS diseases. So now I want to move on to the second story I brought today to talk about the microglia autophagy that is important for clear synuclein and shows the protective effect in the CNS. So the Parkinson disease, as you know, the main hallmark for the pathology, one is the loss of the selective population, namely the dopaminergic neuron in the substantial nigra. And the second hallmark is the accumulation of alpha-synuclein in the Lewy body or Lewy neurites, and then especially the substantial, in many of the different brain regions. Not only in the central nervous system, but also in peripheral tissues, many organs, tissues were found that associated with alpha-synuclein accumulations. And alpha-synuclein is a very abundant brain proteins. There was one study showing that probably up to 1% of total cytosolic protein, brain protein, is synuclein. So this is really abundant proteins in CNS. And the rare mutation of alpha-synuclein has been associated with Parkinson's disease. And then importantly, just the duplication or triplication of the alpha-synuclein and then also some of the SNPs increase the synuclein at a transcription level, that drives the Parkinson disease suggests that the synuclein, just a little bit the overexpression will guarantee the Parkinson disease. This is really a question, the Parkinson disease, about the homeostasis, the alpha-synuclein level in the central nervous system, as well as peripheral tissue. As many of you also know, the Bragg and the colleagues have done thousands of brain pathology, and then they proposed that there was a really spreading of the alpha-synuclein Lewy body from one brain region to another. So the so-called prion-like disease for alpha-synuclein Lewy body. And then also the evidence based on their study, this alpha-synuclein Lewy body could arise at the very beginning at even the guts in the peripheral tissue, and then eventually reach to the brain, and then cause the selective dopamine neuron degenerations. So this is really supporting one of the very important idea that synuclein can be transmitted. First of all, it should be secreted and it can be transmitted, and then spreading out through many brain region and cause this spreading of the disease pathology. And then also the GWAS study for the Parkinson disease, many genes have been associated with the Parkinson disease now. And this work was published about a couple of years ago. They identified 41 PD loci, and the most recent study suggests that more than 90 PD loci that associated with this inherited form of a Parkinson disease. One of the conclusions from this work published 2 years ago, they showed the role of autophagy and also lysosome biology associated with many of the disease risk genes of the Parkinson disease. So we are interested in one of the questions that autophagy is well known to degrade the alpha-synuclein. That's been shown very early on by the group, for example, from David Rubinstein's group, and also David Sulzer with Anna Maria Kilver, and also from Leonardo Stephanes. They show alpha-synuclein can be degraded by macroautophagy proteasome and also chaperone-mediated autophagy. However, the question is the detailed mechanism, how alpha-synuclein is recognized by autophagy machinery, and also the detailed mechanism is not clear. And then that has not been shown in vivo the consequence of the macroglia autophagy, loss of macroautophagy for synuclein aggregation. And then there's no study on the cell type specificity. So we try to address the question. The first thing we did was knock out ADG7 autophagy gene in dopaminergic neuron, which is the most vulnerable neuron in the Parkinson disease. So despite the accumulation of the p-STAGE2 and the ubiquitin inclusion in the dopaminergic neuron cell body, there's no evidence of the alpha-synuclein accumulation in such a young age. However, synuclein inclusion body was identified in a very old mice, 20 months old, at the terminal of the dopaminergic neurons. So this is consistent with the localization of alpha-synuclein as the pre-synaptic proteins. And it is also suggests that the young adult neuron autophagy doesn't really care about much of the alpha-synuclein. And it's probably some of the aggregated or modified synuclein in a very late age or an aging neuron that is important for autophagy degradation. So we start turn our attention to other cell type because a couple of work from especially from Sinjin Li and Nadia Stevanova's group show that not only neuron but also glial cells can reuptake, that can take up the alpha-synuclein secreted in extracellular space and then clear them from the media. This is a very interesting point that we have noticed that, and then we'll start asking a question whether or not the autophagy of the glial cells might be important for the clearance of alpha-synuclein. And Sungjin Lee's actually report that among many cell types that take up the synuclein, the the microglia is the most robust or efficient one. And this is also consistent with the role of a microglia cell as the phagocytes-related cell type that clear off the debris in the extracellular space. So a very talented postdoc, Insub Choi from my lab, start asking questions. So first thing, what he did is to really show alpha-synuclein can be taken up by microglia cells. So what he used is using a transgenic animal models that express alpha-synuclein in a neuron-specific manner. And this transgenic mice was made by Michael J. Fox Foundation, driven by the synapsin promoter, which is a neuron-specific. What he noticed that in my transgenic mice, the microglia cells become somehow activated, where there is a response of microglia to the synuclein expression, which is in the neuron. So therefore, this suggests that the neuron can secrete alpha-synuclein that can be sensed by microglia cells. You can see that the filaments of the lens and also the the points of the microglia cells, they are all ordered in the transgenic mice compared to the wild type. And what he also showing is by isolation of the microglia cells, he can see that microglia cell indeed can take off the alpha-synuclein from the neuron secreted alpha-synuclein. And then he did an imaging showing that the alpha-synuclein localized inside about one cell type, which is the microglia cell specific markers. And he's not satisfied with the transgenic, and he wants to do another model. He injected the AAV also in driven by the neuron-specific promoter, and this is a very robust model, allow the synuclein expression in much high level and a much rapid stage. And then he saw exactly the same thing. The microglia can take up the alpha-synuclein, and then in this case, the alpha-synuclein caused the response of the microglia cell. There is an interesting disease-associated signatures of the microglia cells. This really demonstrate that neuron can secrete alpha-synuclein, and this caused the microglia cells to take up the alpha-synuclein and also respond by changing some of the genes associated with the diseases. So what another autophagy is involved in the alpha-synuclein uptake. And then he cultured the microglia in the Petri dish, and he then treated with the recombinant soluble alpha-synuclein. What he found that the alpha-synuclein, in this case, he used human alpha-synuclein, which can be distinguished from the endogenous alpha-synuclein. What he found that the synuclein formed this inclusion body. And the interesting thing is that he found this inclusion body of alpha-synuclein co-localized with autophagy marker, for example, the LC3, and also co-localized with the P62, and also ubiquitin. And this is he studied the time course. He found that the synuclein can really increase, the puncta can increase over the time, and then the peak at the 6 hour of the treatment, and then gradually decrease over the time course. And this is also the dose-dependent process. The more synuclein you put in medium, the more number of the inclusion body, you can see that. And then he also did the similar experiment, tried to see whether or not this LC3 autophagy marker in vivo in a transgenic model co-localized with the synuclein inclusion in the microglia cells. He found a very similar thing in the in vitro, demonstrate that this indeed occurs in vivo. So to further demonstrate that this autophagy is degradation by autophagy, he performed the electron microscopy experiment, and the synuclein indeed was enwrapped by this double membrane structure, and this is really characteristic of autophagy. And this is also again support that synuclein can be sequestered by autophagy. To demonstrate that such a structure can be degraded by autophagy, he treated the microglia cell that take up the alpha-synuclein with either autophagy inhibitors or the enhancers. So in the case of inhibitors, then the degradation of this autophactome filled with synuclein was blocked. And a taurine, which is an mTOR inhibitor, can facilitate autophagy and also can facilitate the clearance of the alpha-synuclein inclusion body inside the autophagosome. And then he checked the genetic knockout autophagy by knocking out ADG7 and ADG14, specifically in microglia cells. In both cases, the degradation or the PCH2 or alpha-synuclein is blocked. That really confirmed that this structure was indeed degraded by autophagy. I want to point out that disruption autophagy, either by deletion ADG7 or ADG14, has no impact on the microglia survival or overall the morphology in the culture situation. So we are interested in asking the question how autophagy can recognize the synuclein inside the microglia cells. What is of fun is over the time course, there was an increase of a PCH2, not only at a protein level, as well as in a messenger level. This really suggests that the PCH2 level is upregulated by some kind of a signaling in contrast to PCH2 and other autophagy receptors, for example, the optineurin or BR1 and others, has no change when we treat it with synuclein. This is suggests that it was really selective for the PCH2 protein for autophagy recognition of synuclein. And then he knocking out the PCH2 in the microglial cells, and it showed that without PCH2, the inclusion of a synuclein was completely abolished, both at a Western level and also in an imaging level. And then also what he found is in vivo, in the two animal models, especially in the AAV injection, the synuclein co-localized with PCH2 and then was significantly increased in synuclein secreted from the neuron that are driven by the neuron-specific promoter. And the PCH2 and the synuclein can be co-localized very well in the microglia cells. So in order for the autophagy or receptor to recognize synuclein, and we hypothesized that PCH2 should interact with the microglia, with the synuclein. So what he performed is the immunoprecipitation in two animal models as well as the microglial cultures. In all the cases, he can see there was an interaction of alpha-synuclein with the oligomer form of the PCH2. And as we know that PCH2 oligomerization is important for the cargo recognition and also for the sequestration. So this result really support that there's possibly a direct interaction between the receptor PCH2 and the alpha-synuclein that sequester synuclein into the autophagosomes. So how does the synuclein get inside the cells that can be recognized by PCH2? So we went through the literature and we realized that a couple of work published again by Sung-Jen Lee and then Stevanova's group, they suggest that the toll-like receptor 2 and possibly toll-like receptor 4 can sense the alpha-synuclein. And what they show is these interaction could drive this immune response in a microglia cell or astrocytes. Then we ask a question whether or not the toll-like receptor 2 or 4 or maybe others can signal to the microglia cell and drive the PCH2 upregulation inside the microglia cells. And then, as you know, that many toll-like receptor was found in glial cells, including astrocyte or microglia cells. And then we will ask a question whether or not we want to just screen all the major toll-like receptor and see whether or not any of these are specific for alpha-synuclein interaction and drive this signaling enough we use one of the reporter for these toll-like receptor based on the I-kappa-B or NF-kappa-B drive luciferase assay to study the interaction between the toll-like receptor and alpha-synuclein. So the work done by a student in the lab, Steve Segovin, and together with INSAP, so they utilized the luciferase reporter that sort of sense the toll-like receptor. So they and then they use the HAP293 T cells, which do not express any toll-like receptor. So they then reconstituted the receptor by transfecting one by one and co-transfecting with the luciferase reporter. And the first thing that what it did is to show that system works in the hand, and then they transfect either toll-like receptor 4 or 2 or 5, and they use their agonist to treat the cells, and then they can see that in each case the toll-like receptor and can activate downstream the NF-kappa-B signaling and drive the luciferase activity. So having set up the system, then they ask a question, what happened to synuclein? And then in the case of a toll-like receptor 4 complex, and then they can see clearly there was a dose-dependent synuclein interaction with the toll-like receptor 4, and also in a case of a toll-like receptor 2. And this is really consistent with the previous work from Sungjian Li and Stevanova's group. And these indeed can interact with synuclein. And which one is preferred or the dominant? So we then co-transfect 2 and 4 toll-like receptor, and then blocking the toll-like receptor 4 with the antibody, and then it's treated with alpha-synuclein. And in such a case, we found that the toll-like receptor 4 actually is more important for, or at least preferred for the interaction with alpha-synuclein based on the luciferase assay. And then we went back to the microglia cells and taking the toll-like receptor knockout and then treated with alpha-synuclein. So, and then clearly we saw that the p62 expression was abolished or at least compromised when we treated with alpha-synuclein at both the protein level, also at the messaging level. And also we treated with the toll-like receptor, the antibody that sort of abolished the activity. In such a case, we saw the similar thing as we used the toll-like receptor knockout microglia cells. There was also the downregulation of the p62 expression. And importantly, in the toll-like receptor knockout microglia cells, the p62 level or co-localization with the synuclein was also reduced, providing a strong support to the role of the toll-like receptor that important for driving the p62 expression and then sequester alpha-synuclein inside the microglia cells. So one of the very important agonists or ligand for toll-like receptor 4 is the lipopolysaccharide, as you know, the LPS. And the LPS is probably the most well-characterized ligand for stimulating toll-like receptor 4 and driving the immune response. So then we ask a question, we're curious about just to compare alpha-synuclein and LPS, whether or not they show similar way for immune response and also p62 expression. So I want to just make this a long story short. We found that the only thing that in common to these two between synuclein and LPS is the upregulation of p62. In the case of the other gene, interleukin-1 beta, and the TNF-alpha and others, these are important for immune response. The LPS much robust, 100 times higher than synuclein. This is shows the difference between these two ligands in the case driving the immune response. And also one of the very important protein, the IRF3, the phosphorylated form, and it's only was found in the treatment LPS, but not in alpha-synuclein. And it shows the difference between these two ligands in terms of TL4 interactions. And we also know that TL4 responds to the LPS by endocytosis and the signaling inside the cells for the immune response. And we found that LPS indeed can cause the endocytosis by this fast sorting. However, the synuclein cannot induce the endocytosis. And again, the case we treated the endocytosis inhibitor, the dinosaurs, and that has no impact on the synuclein induced p62 level. And imaging level, we had no evidence of synuclein co-localized with an endosome. This really demonstrates that, unlike LPS, synuclein do not cause endocytosis of the TL4 receptors. And what's the downstream of the TL4? And one very important signaling is NF-kappa B. And I want to make a long story short that we found that indeed the NF-kappa B signaling is downstream of the TL4 signaling, the kinase, the receptor signaling. When we block the NF-kappa B using certain inhibitors, we can abolish the p62 upregulation. And interestingly, synuclein can also cause the MAP kinase signaling. With the MAP kinase inhibitors, there's no impact on p62 upregulation. And then we showed that the toll-like receptor knockout can abolish the immune response in the microglia cells. And this, again, really demonstrates that NF-kappa B confers the alpha-synuclein toll-like receptor signaling in the induction p62 in microglia cells. So, okay, so I just told you what we found mostly based on the culture, microglia cells. What's the consequence in vivo? What's the significance of the microglia cell clearance of alpha-synuclein? So what INSUP did is he created a conditioned knockout mice, deleted ATG7 from microglia cells, and then he crossed with these alpha-synuclein transgenic mice, which overexpress synuclein over endogenous 2 or 3-fold, which really to mimic what the genetic disease, which in the case of alpha-synuclein expression, triplication for the Parkinson disease. So what happened is we did not see anything degeneration or microglia cell death or neurodegeneration in ATG7 condition knockout microglia cell only, or the alpha-synuclein transgenic mice. So in that case, what he found that there's really accumulation of alpha-synuclein in insoluble fractions in both AAV synuclein injected case as well as the transgenic animals. You can see there was clearly a smear of alpha-synuclein in an oligomer form compared to the control mice. And the same thing in the transgenic mice. And in the imaging, so what he found that when he removed the ATG7 from microglia cells, there was an accumulation of alpha-synuclein in the microglia cells. And also in the dorsal substantial nigra, the dopaminergic neuron, there was really increase of those puncta of alpha-synuclein. And also in as well as the dorsal striatum. And this is to demonstrate that without autophagy, the synuclein can accumulate in the brain and forms those inclusion bodies. And interestingly, and consistent with the protective role of microglia cells, when we put these two together, synuclein transgenic, and remove the ATG7 from microglia cells, there was a degeneration of the dopaminergic neuron, specifically in the substantial nigra. And it was shown by both the TH staining as well as the nasal staining. And this really demonstrates that microglia autophagy as a neuroprotective. So to summarize, what we have found is the microglia cells, unlike the neuron or other cell type, we think that the synuclein can interact with the toll-like receptor, peripherally with the toll-like receptor 4, and then stimulate there's a signaling of a kappa B and then drive the p62 upregulation. And the p62 can recognize the synuclein, most likely through the ubiquitination of the synuclein, and then sequester them in the olfactome and degrade them in the lysosome eventually. And this is in a contrast with the LPS, because the toll-like receptor interact with alpha-synuclein does not cause the endocytosis of the toll-like receptor. And in a contrast, LPS can cause this endocytosis. And the signaling, the inflammatory thing, which is the absence in the alpha-synuclein case. When we block this autophagy, and there is an increase of alpha-synuclein inside the neuron, as I show in the animal model study, and then drive neurodegeneration, for particularly in dopaminergic neurons. Okay, so I would like to acknowledge the people who did the work. This is the guy, very talented postdoc, Insub Choi, and students, Stephen Singobin, they mainly responsible for the microglia autophagy clearance. And also Zichang Deng, who did the work in P62 ALS mutation. And Shenting Li and others help with the work in the neuron culture and also the animal models. Many people I'd like to also acknowledge, and collaborators and the fundings, and thank you very much. Questions?
Hi, really interesting story. So if it's not TLR4, what do you think is mediating the actual ingestion or of synuclein?
Yep. So that's a very important question. So what we have shown you is that there's definitely interaction based on our genetic data and also antibody blocking. Synuclein interact with TL4 receptors. However, we have no idea how synuclein get inside the cells. So based on many people's study, synuclein can really freely penetrate inside the cells without even receptors. There is evidence from both sides, receptor mediated and probably mostly oligomer form of a synuclein endocytosis getting inside of the cells. There was also a receptor independent process of a synuclein can get into cells. So in our case, we think, and then Insub can also jump in. In his study, what he found that the synuclein can rapidly get in and also can come out quickly. So there is to us, this is really a sort of endocytosis role receptor independent process. So what we think is that the receptor can interact with the synuclein, rapidly induce the signaling, but synuclein just get into the cells without toll-like receptor mediated endocytosis.
Hey, hi. Great talk. So I have a question for you. Has this been studied in a particular ethnicity, let's say Ashkenazi Jews, and how does this vary and how does this span out in a bigger picture?
Can you repeat it again?
So has this been studied in a particular ethnicity, as in, let's say, in a particular ethnic group?
Particular.
The Ashkenazis have differences in microglia.
Oh.
Yeah.
Okay. Yeah, I heard that actually. All right. I don't know. So the pathology analysis is, which there's still a paradox we try to understand. So there is a really beautiful pathology by groups in Australia. They show the pathology of a synuclein in astrocytes, of course in neurons. And so far there was a little evidence on the microglia autophagy accumulation. So one of the arguments we had is maybe microglia just really quickly degrade them and they digest it, and it's very hard to catch them. Yeah. So the question is, I don't know for whether or not in different ethnic groups those pathology is different. And then you mentioned about the Ashkenazi Jewish population, the LOC2, right, mutation.
Exactly.
maybe that's what you are intend to ask a question. So we have tested. I think my students here can really help me out. So we have a culture, the LOC2 mutation, the G2019S mutation, as well as the knockout. And we have seen there's a reduction of the synuclein inclusion body formation. So we're not so sure it's due to more clearance or reduce of the autophagy function. So that, because that related to both way. So we have yet to find out that case, but at least we can see there was reduce the autophaxone or inclusion of the synuclein inside the microglia cell, lacking PCH2 or the mutants of LOC2.
Thank you.
Yeah.
So synuclein can basically cause widespread microgliosis. Do you know if any of that downstream activation is dependent on autophagy? So if you knock out autophagy, do you still see activation of microglia if it's unable to process the synuclein?
Yeah, that's a really good question. I don't think we have looked at that carefully, synuclein, but in the case of role of autophagy in immune response has been well documented in macrophage or some other immune system cell. But there was very little known about microglia cells, and we are really looking at that now. Whether or not autophagy plays any role in microglia cells, immune response. I would be surprised if there was no change. I think there might be a change in that. Yeah.
I ask, so in the SOD1 model for ALS, if you knock out autophagy selectively in neurons, we see a reduction in gliosis. I don't know how these two things would be related, but maybe autophagy somehow is tied to inflammation.
Yeah, that's a good question. So we have not done an experiment. That's very obviously the thing where people can do with a lot of ALS model, cross with autophagy, compromise the mice overexpression. Particularly, I think, the PCH2 that has been studied by some groups showing some change in TDP43, as well as other, maybe another model for ALS. There's an aggregated SOD1 model. I think there's some study over there, but I don't remember the result. But it's a very important question to address. We really believe ALS has a really important components that might disrupt some of the autophagy components. And then interestingly, C9orf72 expressed a very high level in microglia cells that has been shown. There was a lot of interest in study ongoing. So yeah, it will be interesting, that question. Yeah. Okay, so thank you. We'll move to
Okay. Oh boy, do I have to repeat everything I said? So everything we've been talking about so far is macroautophagy, right? Let's not forget these amphisomes when we're talking about neurons. Nobody talks about them. The endocytosis is occurring all the time in neurites, all the time. And many of what we're talking about is autophagosomes really are endosomes that are fusing with local autophagosomes and moving together and eventually fusing with the lysosome. Now, we've been studying this macroautophagy because of these nice tools, LC3 and knockouts, which I'll mention specifically in a minute. But there are other types. Ana Maria Cuervo has been working for many years on chaperone-mediated autophagy. This is probably very important. It could be very important for Parkinson's and the sort of things that Zhenyu was speaking about. Microautophagy is pretty interesting. Been very little work on it, but those of you who have looked at electron micrographs of neurons know that these structures are extremely common, especially presynaptically, also called multivesicular bodies. And then there's another type, these so-called multilamellar bodies. There might be only, to my knowledge, only 1 or 2 papers in neuroscience that even mention multilamellar bodies, yet they are seen in virtually whenever you do look at a pathological specimen from a disease, you're going to find these structures. So these are very important structures, and they're independent of LC3, and we don't know how to look at them very well, and they're just simply not studied that much. Okay, so if I had time, I would go through these different steps. So all 3 speakers today have depended on this group, Komatsu et al., this is one reason we study this so much and so well, is because they introduced a way to remove ATG7, another group ATG5, specifically from specific neurons back in 2006. So I and Zhenyu both used that in microglia. We were early adapters, so we started using this to knock out autophagy developmentally, of course, from dopamine neurons, from cortical neurons, now from medium spiny neurons. People often complain that the professors are giving talks which are outdated, it's not new stuff. But in fact, this paper came out, it's on PubMed now, because I was looking at, it wasn't on PubMed 2 hours ago, because I looked it up because I was preparing the lecture. And while Zhenyu was there, I got on to is Bill Murray here? Did you see that if you look for servers, Bill Murray's server comes, there's 2 Bill Murray servers. Anyway, you have a guest genome center server, and I got on, and Ori's new paper is now on. So that was just on in the last half hour. So this is new stuff. You can't download the paper yet, but at least you can read the abstract on PubMed. So that, and then there's a review of all this stuff, and that was just online yesterday. So this is between, some of this stuff is a day old, and some of it's half an hour old. So anyway, that's autophagy in the striatum, and for the interest of time, okay. Not for the interest of time. Who cares, right? We got why were we interested in developmental disorders? This is pretty interesting about, classic what we used to call autism, ASDs now, schizophrenia. There's a bunch of genes that if they're deleted or if there are extra copies, they will give you either autism or schizophrenia. Okay, and there'll always be, one of them deletion and one of them an extra copy, but it can go in either direction. So this is quite suggestive that there's something going on in development that could flip. And then looking at what are some of these signals? Well, here's a list of some of the signals. There's now over 100 autism genes, and when I go to the Simons Foundation, I asked one of the geneticists, "You mean really 100?" He said, "Well, actually, we're over 1,000 now. We only have 23,000 genes. 1,000 of them apparently can be involved in ASDs." But let's look at some of the ones which are monogenic and a little more clear. So here's some of them. And in particular, let's talk about tuberous sclerosis, which is, most people with tuberous sclerosis are not autistic, but a high number can be, and epilepsy shows up in a large number of people with autism. But one thing that unifies many of these together is they all regulate mTOR. They're all mTOR regulators. So why should anybody care about that? Well, I'm not going to go through all these signals now because it's now 6:51, which means officially I have 4 more minutes. So, if you look at P10, you look at the tubular sclerosis, you look at neurofibromatosis, they all will work on this mTOR pathway. And as both previous speakers already alluded to, mTOR pathway is a regulator of autophagy. So the only thing I think you're really going to have to remember here is that rapamycin is a way that you can kind of skirt all this stuff. You can inhibit, no matter what you have with tuberous sclerosis up here, if you have rapamycin, you can turn down mTOR and you can activate autophagy. I know I'm simplifying this. So a whole bunch of autophagic dysfunctions at this point now implicated in these disorders. And I'll just mention again, if you want to read hot off of the press, it says 2019, but in fact it's from yesterday. So that if you read this review, it goes into great detail on that. Ori's review, Ori just graduated about 2 weeks ago. Got his PhD about 2 weeks ago. This is just some of them, again, because of time I'm going to skip this, but this is sort of the existing literature right now. There are changes in presynaptic release, some receptor degradation, synaptic plasticity, synaptic structure, and some of this actually came from I's group and Zhenyu's group. Anyway, so early work from our group, Carolina Cebrian and Daniela Hernandez, Ciara Torres, they happen all to be from Spanish-speaking countries, and looking at the dopamine, knocking out ATG7 out of dopamine neurons. I'm just not going to give you the details on this. I'll show you that you can, with rapamycin, you can get large autophagosomes. You can get stuff inside of them that looks like synaptic vesicles. And in fact, the terminals are too large in a knockout with dopamine, autophagy knockout in dopamine neurons. This does not happen in all neurons. So one of the themes here is that if you remove autophagy developmentally, it's different from neuron to neuron. So here they're too big. If we knock it out of striatal neurons, one of the classes of striatal neurons, the neurites are too small. We don't really have a good explanation for that. I'll skip this is autophagosomes eating up synaptic vesicles. I'm going to skip that and just say you can regulate the amount of neurotransmission. So you can measure dopamine release. This is a technique called cyclic voltammetry, where you could directly measure dopamine as it's being released. And you can see if you have a knockout, you release too much, which is maybe not a surprise given that you have more synaptic vesicles. But if you add rapamycin, you can get an inhibition of release, but not if you have a knockout. So anyway, there are presynaptic effects on synaptic vesicles, and they get into neuronal plasticity. And if we had time, I'd go into it. Let's go more into the cortex, because for the people interested in autism, that's going to be a little more interesting. So what happens during normal development is, this comes from a fellow named Peter Huttenlocher, died 2 or 3 years ago. And he would this is where he got his data from. He would look at electron micrographs of human brain tissue. So human brain tissue has really bad morphology, because you can't even take a department chair or a dean and fix them with glutaraldehyde by perfusion. I know we'd like to do this. Where is Dr. Maniatis, by the way? Anyway, so we have, I know it's jumping back and forth without me being even close to it. So what survives? These synaptic densities survive. They survive even though everything else is getting proteased up. So he can sit there and count them. So poor Peter Huttenlocher counted the numbers of synapses in the cortex from people who died at these different ages. And you can see what happens here. Well, from the age of 10 to 15, you get an increase in this part of the cortex, and then you get a decrease. So you're always making and unmaking synapses all the time. You have a net making in relatively early development, and then you have a net pruning over time. And this is a nice paper that looked at different parts of the cortex, and we have techniques now that allow us to be much more lazy than Peter Huttenlocher was. And again, we can look at ages and see there's an increase and then a decrease. Now, we can look at the synapses now in autism and see if there are any changes. This was a theory, and this is all work from Gaomei Tang, who's now an assistant professor at Columbia, when she was a postdoc. And so this is Golgi staining of human tissue, and you could see these nice dendrites over here. In fact, they're so nice that let me show you the three-dimensional. Even though I think my time is officially over, I'd still like to show these cool movies. So this is human. This is a human dendrite from a Golgi stained. From I think in this case, a boy that was about 15 when he died. And you can sit there, you can count the number of spines, and that is the number of synapses. You can probably see from here that, well, I'm not going to ask you, again, just because of interest of time, but this is an autistic child who died at 15, and a control child over here. Can you see the differences? So this is giving us the impression, and we quantify this, that there is not a net loss of synapses in this part of the cortex, this temporal cortex, during maturation with autistic children. And here are the numbers. So these are ages. So this is what happens with control. So kids starting at 2, 5, 6, 8, and then 18, and you can see there's this loss. This is not happening with the autistic kids. Now, this is just one part of the cortex, and there are going to be differences, not just between neuronal types, but different neuronal areas. So this is the change in numbers of synapses per unit length in normal kids, in our normal development, and this is what happens with autistic kids. We can also do this with Western blots and measure the pre- and post-synaptic components, which, for the interest of time, I'm going to skip. We can also look at P62, which you've been hearing about from Zhenyu Yue and from and also from I. And I'm just skipping through this because of the interest of time. Let's go to the stuff that's going to be the most interesting for you. So less synapses. The mitochondria are building up in the autistic kids. We're getting much more mitochondrial density. These mitochondria are sick. We can use, for instance, we can look at oxidized guanosine to look at the mitochondrial DNA, and it accumulates. So something going wrong, not enough sufficient autophagy in these neurons. This paper, if you want to read more about it, Gaomei wrote a paper going through all this. So summarizing the human stuff, ASD patients have higher numbers of damaged mitochondria, and they have higher synaptic density. This, by the way, around the age of 40 starts to even out. So there is a second synaptic loss that starts to occur in older people with autism. So let's look at mouse development. So we did all this in mice, but with mice we can get these beautiful, there we can fix, and we can really count the numbers of spines very nicely, and we can look I'm doing this so quickly. But if we look in an autism mouse model, this is a tuberous sclerosis mutation. Normally, we get the loss of synapses, but instead of over the course of age 10 to 15, we get it in the 3rd to 4th week. So this is what happens with normal mice, but with these, why does it do this? With these autism model mice, we start getting, we don't get a net loss. We can, and just to save time, I'll say, if we activate autophagy using rapamycin, which you'll remember is a way to do this with drugs, we are able to retain we're able to get the normal loss. So I'm going to skip all this and just say, if you want to look at the paper, Tang et al., Neuron 2014, you can get all these many details, and you can see that there is, in the cortex, a normal loss of synapses, which is dependent upon macroautophagy. And it's definitely macroautophagy, in this case, in the cortical neurons, because using those Komatsu ATG7 Cre knockouts, this is only in cortical neurons. It's not in microglia. It's not in interneurons. Okay. So we can, oh boy, there's so much stuff that I would normally show. And this is all wonderful work by Gaume, and you just simply can't hear it. And this even works for social behaviors, and we can normalize the social behaviors. We're just moving on. So the last thing I have, because I really am out of time, is a new paper. This is the new paper that came out in the last hour. And this, again, it's from Ori Lieberman. The idea here, I can tell by the questions here that a lot of the people are neuroscientists, so I don't have to go into this too much. For those of you who don't study neuroscience, forgive me. Normally I would tell you more. But we're going to look at the neurons of the basal ganglia in the striatum. These are the neurons that are called direct pathway neurons, indirect pathway neurons. These are the neurons which kind of integrate your stimuli and what you already know how to do and tell you what you're going to do next. And so we can remove autophagy using the same trick, ATG7 and Cre, out of direct pathway or indirect pathway neurons. These are very difficult neurons to tell apart. You normally can't tell them apart. You can't tell them apart by looking at them. You can't tell them apart by electrophysiology. Yet they have very different responses to autophagy knockouts. So let's knock it out. So so this is a little similar to cortex. When you're young, if you get any stimuli at all, somebody yells at you, a light goes on and off, whatever. We know this from mice, obviously, and from monkeys. When you're young, that will cause these neurons to fire. They'll be very active, okay? But when you get older, and older in the mouse means around P28, and human probably, again, means around, 7, 8, somewhere around there, they become much harder to drive. They become very much less active. Why are they less active? I'm going to lead a bunch of slides right now and just get to the point. It's because they start expressing high levels of a particular ion channel, which is a potassium channel. So if you remember your neuroscience courses, when you open up potassium, that allows potassium usually to leak out of the cell and hyperpolarize the cell. And this allows the cell to be less excitable, right? That's how you end an action potential. You open up potassium channels. Now, this is a special kind of potassium channel called an inward rectifying channel. They're sometimes called Kehr channels. Kehr channels become activated as these neurons mature. They're a funny channel because they don't open up when the cell's very depolarized. They mostly open up when it's hyperpolarized, and that tends to clamp the cell towards the potassium reversal potential, which tends to be around -70 or so. So it keeps the cell really silent. And so you need a lot of activity in order to activate. You need so much cortical and thalamic input that it drives it, you drive from this near the potassium reversal potential all the way up here. And this is called an upstate. And now that you're in the upstate, you can have action potentials. You go back to the downstate, and the Kehr is holding you here, and no matter what you do, you can't get that neuron to fire, okay? So that's what's being regulated. I'm going to skip all this and say, look at the paper that came out today, and you're going to see that what's being regulated is this Kehr channel. This has behavioral consequences that are going to go along with autism and related developmental disorders. You could see the differences in the behaviors over here. This is motor learning behavior. The motor learning behavior happens if you knock out autophagy about either. But the point is that you get different responses if you remove the autophagy from the direct or indirect pathway neurons. The direct pathway neurons, which are the neurons that tell you to do something, to go, they're classically called go neurons. These, if you knock out autophagy, you get fewer spines and you get less dendrites. Does this make any sense? No, it doesn't make any sense at all. It's the opposite of what I just told you about the cortex. This is why we're going to have to, if we are studying these changes, we're going to have to go neuron to neuron and time to time during development. Okay? Indirect pathway doesn't care. There's no differences in spines, there's synapses, there's no difference in the morphology of the neuron. But what they have is they never become less excitable. They never mature. That is like the cortex. Without autophagy, you don't get normal maturation. And too many details here for a time, but the resting membrane potential doesn't mature. The input resistance, which tells you how much voltage you're going to get per amount of current that you inject, never matures. Riobase is an antiquated name, which means how much current you have to, how much current you have to give to a neuron before it fires. These details I'm not going to, again, I'm sorry, just simply not going to get into. But for those of you who do electrophysiology, you will recognize this. We're giving changes in the voltage over here. We're measuring the changes in current. And these changes, the difference between this and this is that fancy potassium channel that I told you about. So you get much less of that potassium channel activity if you knock out autophagy. Now that doesn't quite make sense either, but Ori Lieberman figured it out. So what he does is, I will skip this to show you. There is, in fact, let's just go right to the punchline. This channel is not being degraded. So this would be a normal channel. This is the degradation of a normal channel. If you knock out autophagy, you can see there's no degradation. Internalization, that endosomal stuff I was advocating for everybody to study when they're looking at this pathway, that's normal. But it's later, probably when it's fusing with the autophagosome. That's where things are going wrong. Now, why are they going wrong? It's staying on the surface, and yet you saw the activity is less. So there's more there, but it works less well. How can that be? And that's where Ori's post-translational modification hypothesis comes in. The idea is that you would get something like ubiquitin, but I'll show you it's actually acetylation, which modifies on the cytosolic side here. And when that happens, you inactivate the channel. It still comes in, if you inactivate the channel, normally it will probably fuse with the autophagosome, so the arrow should probably actually be here. Well, it is here. And you get an amphasome, and that amphasome fuses with the lysosome. So that's what we think is happening. Now, how did he test it? The way he tests it is very clever. First, he looked at ubiquitin. There's virtually no change in ubiquitin, but you could see with the knockout, there's a big change in acetylation. Acetylation is always on lysine residues. And he said, okay, what are the lysine residues that could be acetylated? Now, here's these potassium channels are, I'm sorry, these KIR channels are in all these different kind of organisms. They're in flies, they're in worms, but these lysine residues are maintained. You can mutate the different lysine residues. There's one lysine residue, it happens to be this one, that if you modify it to asparagine, so you can't put a lysine on it, then you can't turn over the protein anymore. Okay, so that's our culprit. So it's this particular lysine. And so what happens is the lysine will, if you so normally if you knock out autophagy, as I showed you before, I know this can be a little hard to read. So this is the normal potassium current. This is the potassium current in the knockout. Remember, it's much less. But if you make this cure so you can't acetylate it, then you never get this loss. It just keeps hanging out. On the other hand, if you make an imitation lysine, you're able to get some. You're able to remove the fake acetylated KIR2 from the membrane, and now you can get a normal reduction. Okay, I know this is all super fast. So that's pretty much it. So the conclusion: novel role for autophagy and regulation, and that's on channels now. So that adds yet another change that can occur in synapses. And this also brings up, so acetylation, like Huntington, for instance, has been known to be acetylated for a long time. People have suspected that acetylation, this is more for the cognoscenti, as well as ubiquitination, can also be a marker to say that this protein should become, undergo autophagic degradation. So this is additional evidence. This is another signal we should be looking at. So I just wanted to say these are all the, this is kind of what's in the literature. You can read about it in this new review. And now we have to add specific clearance of membrane proteins. So likely receptors directly from the plasma membrane and certainly channels can be turned over. This is not obvious, right? Because normally you would think of macroautophagy working on cytosolic soluble sorts of proteins, but can happen also on membrane proteins, probably because of this important endosomal intermediate. So the endosome takes in membrane proteins, and now this can go through the through autophagy cycle. Okay, so that again, I would have normally done that in about an hour. I know I went over a bit too long, but I hope not much too long. So the people I just want to mention again. So Ori wrote this new paper, and just graduated 2 weeks ago. Gaomei Tang, Daniela Hernandez, and Ciara Torres, who worked on the dopamine system and the cortical system. Thank you very much. We have about 15 seconds for questions. Yes.
Hey, David.
Oh, you can't ask questions.
I can ask now. So are you saying that the potassium channel acetylation is a signaling for autophagy degradation, or the opposite?
The acetylation is apparently a signal for autophagy.
Yeah. That's interesting, yeah.
Yeah. And so there's going to be a P62 or optineurin or something like that that's going to recognize that and bring it in. But the rules are going to be different because it's an endosome. It's going to bring it in as an endosome. I've talked you all out, or else you all want cheese and crackers and wine. Yes? Rapamycin is given for tissue rejection. So when people are given transplants, for instance, they're given rapamycin. So it is toxic, and it's also given to hundreds of thousands or millions of people. I'm not advocating giving rapamycin to children who are diagnosed with autism. However, I don't think we should completely dismiss using mTORCI. I think Alexa's listening, and Facebook just decided now that we're talking about autism, I should be censored. So I think the problem, we can argue about this, including over the beer that I understand they have waiting for us, that what we're doing is lumping autism together. Remember, there are somewhere between 100 and 1,000 genes that are causing autism. And I think for patients who are working with insurance companies, it can make sense to call Rett syndrome and Asperger's syndrome and TSC and all these different things all autism. However, I think if we could go ahead, if we could look 50 years from now, and the future scientists will think that we were incredibly primitive and kind of stupid to lump all these together. Now, if we don't start separating these different forms of what we now call ASDs, I do not think that rapamycin or actually any drug treatment is going to work, because what you're going to do is say 25% of autism is mTOR-related autism, right? You give them all rapamycin, it can only work on 25% of them. There's absolutely no way that it would ever pass. People would say it's a failure. And in fact, rapamycin has been tried in small, mostly for tuberous sclerosis, and so far has not been successful. But we have to wonder, is part of this issue, not the whole issue, is the lumping. In my opinion, the most important of all those is stratifying the type of autism. I think I would really like it if we can, sooner rather than later, stop talking about autism. I understand that there are political and insurance reasons to lump autism, but I think medically this is a big error. So we have to come up with biology. You have this flavor, and you can still call them autism if you want, but you have vanilla, you have strawberry, and we need different flavors. Okay. Thank you very much.